There are some salts which are even used, like if we will use the high calcium concentration in the range of 12 millimolar concentration, that also help to stimulate protoplast survival and cell division. That help to stimulate protoplast survival and cell division by the addition of the higher concentration of calcium in the medium. And the another effect of ammonia has been observed. They have observed that if we increase the concentration of ammonia greater than 2000 milligrams per liter, that harms our protoplast. So whenever ammonia as a nitrogen source, when we have to add this ammonia as a nitrogen source in the protoplast medium, that time we have to be cautious that its concentration should be less than 2000 milligrams per liter. Otherwise, this will kill our protoplast. So these were little bit about the protoplast culture medium conditions. Now the main which we have to discuss is the regeneration method, or how protoplasts are regenerated. In our previous lecture, we have discussed about the protoplast isolation and the protoplast fusion. So third topic was there, protoplast regeneration. We have to be sure that the protoplast which we are using to regenerate is in proper ratio. That is means their concentration in the medium should be appropriate. Otherwise, these protoplasts will not be able to generate cell wall and grow. So we can see their density by with the help of hematocytometer. We can observe or see their density. जी बेटा, वालेकुम सलाम. अच्छा सॉरी, आप लोग अब एंटर हुए. आप लोग क्यों लेट? हाँ, आज कुछ इंटरनेट में इश्यूज है बेटा, मतलब इवन मैं भी एक ज्वाइन मीटिंग नहीं कर पाई. टुडे वी आर डिस्कसिंग अबाउट protoplast regeneration. In previous lectures we have discussed our protoplast isolation, then fusion. Now we have in our hand, we are having fused protoplast. Now we have to go for the protoplast regeneration. In protoplast regeneration, we have to be sure that whatever protoplast we have to generate is in appropriate density. The density of these protoplasts are determined by using hematocytometer, so that we will be able to maintain the proper density of hybrid protoplast. So we are having 7 different types of regeneration method for protoplast cultures. First is the Agarose embedded cultures. Second is liquid droplet culture. Third, culture on feeder protoplast. Fourth, double layer method. Fifth, agarose beads. Sixth, microisolation. And the last one, seventh, co-culturing. So first is agarose embedded cultures, liquid droplet cultures, culture on feeder protoplast, double layer method, agarose beads, microisolation, and co-culturing. So agarose embedded cultures. So we have to take our fused protoplast on the agar medium, which is semi-solid in nature. This agar should be in a pure form. The agar which we are using for protoplast regeneration should be in a pure form, and it should be able to solidify at low temperature. That is, low gelling temperature agarose we have to choose. The type and the concentration of agar matters. The type and the concentration of agar matters. First, we have to take purified form. Second, we have to take concentration 0.6%, so that the medium which we are using for protoplast development should be in a semi-solid form by using 0.6% of agarose. So it has been observed that there are some protoplasts which are heat sensitive. So that time we can't use for them, we can't use simple agarose. We that time we use for heat sensitive protoplast, we use alginate. We use alginate as a solidifying agent. So the heat sensitive protoplasts are mostly taken from the Arabidopsis thaliana. The heat sensitive protoplasts are from the Arabidopsis thaliana. When we are working on the Arabidopsis thaliana with its protoplast, that time we can use alginate as a solidifying agent. So when we place our hybrid protoplast on the agarose, then we can seal this agarose with the parafilm, so that only there will be gaseous exchange, but it will be free from the contamination. When we observe that there is appearance of small colonies on the semi-solid medium, that time we can cut the agar along with the protoplast, then place this agar along with the protoplast in the liquid medium, so that this protoplast will develop into the callus. First they will form the cell wall, then from cell wall to they will form the callus. But we have to be sure and specific the medium which we have used in case of semi-solid media, same media we have to use it for the liquid media. Means we can't change the media because it's the fused protoplast. Or in earlier cases we have seen that whatever media we are using in case of solid media can't be used in a liquid media, but in case of protoplast development we have to use same media in solid media as well as same for the liquid media. So this was Agarose embedded cultures. So the second is the liquid droplet culture. Second is the liquid droplet culture. What we do, we take our protoplast, hybrid protoplast, with the pasture pipette, zero point 0.1 to 0.2 ml in the form of 0.1 to 0.2 ml droplet. We take this protoplast in a sterile petri plates. The size of the petri plates vary. We can take 3 centimeter petri plate in diameter, or 5 centimeter dia, or 9 centimeter dia. When we take this protoplast, then we add few drops of nutrient media to it, then we seal these petri plates with parafilm, so that there will be no contamination. So this is a simple method, like micro droplets are there in the petri plates. It has been observed that the protoplast development in liquid droplet culture are having many advantages. First, there are some species of protoplast which are not able to divide on the semi-solid media. There are some species of protoplast which are not able to divide on the semi-solid media. For those protoplasts, we can use liquid medium. So this was the first application. Second advantage of this is that we can effectively reduce the osmotic pressure within in the few days of culture, which this osmotic pressure is created by the use of osmoticum. At the first stage, when we are adding our protoplast culture, which is without the cell wall in the medium, that time we are adding higher concentration of osmoticum. As the within in the 24 hours, this protoplast is able to generate cell wall. So it means that within 24 hours we have to decrease the osmotic pressure of the medium by introducing fresh medium into it without adding of any osmoticum to it, without any addition of osmoticum to it, so that there will be decrease in the osmotic pressure. This technique can't be applied to the semi-solid media. The third advantage of this is that when protoplasts are able to generate its cell wall, then they will be able to form callus in the liquid media. So the density of the cells in the liquid media will get increased. In case of liquid media, we can take these small clumps, then place these small clumps into the fresh media. So it means we can maintain the cell density in the liquid media. Density of the cells can be reduced. Now the fourth important point is that if we see there are some release of phenolic compounds in the media, or if we will see there is a color change in the media, that time we can change the media. We can change the media. That is not applicable in case of liquid culture. So this was about the advantages of the liquid droplet culture. So the third is culturing of our hybrid protoplast on feeder protoplast. Third is our culturing of feeder protoplast onto the culturing of our viable hybrid protoplast on the feeder protoplast. As you have studied that in embryo rescue technique, what we do, we take hybrid embryo and we allow it to grow on the endosperm. This endosperm is also called as nurse cells, or we can say this endosperm act as a feeder cell there, which feeds this hybrid embryo. In a similar way, here also we can use feeder protoplast. We can use feeder protoplast which help our hybrid protoplast to generate cell wall and to be in a viable condition. To this feeder protoplast cell, we X-radiated it so that this feeder protoplast loses its ability to undergo cell division. These will be only the viable cells. They won't be able to go for a cell division. So on this feeder protoplast, we will place our hybrid protoplast so that the viability of our hybrid protoplast get increased. So the viability of protoplast get increased. So that also increases our plating efficiency. So this was about the feeder protoplast. If we talk about the double layer method, in case of double layer method, as the name indicates, we have to add agarose 2 times. First, we take a sterile petri plate, then we make a base of agar nutrient along with the nutrient media, agar. Then we place our protoplast over it. Then again we will pour freshly prepared agar along with the media onto it so that the protoplast get embedded into the agar medium. So these protoplasts get fixed up in the agar medium. So these are able to generate and they are able to form cell wall there only. When they are able to form cell wall, when they are able to generate, but they don't have that much of space to divide more or to form the callus because there is overlaid with the agar. So what we will do, we will cut agar along with the agarose. Then we will cut this agar along with the protoplast, then place it onto the liquid medium in the shaker incubator, where we have to maintain temperature within the range of 25 to 30 degrees centigrade. So then this agar will be able to form the callus. From callus we will be able to generate plants. So this was about the double layer method. So fifth is our agarose beads. In case of agarose bead, what we do, we take our hybrid protoplast, then coated it with the agarose and pour this drop into the liquid medium. As we know how to immobilize the cells. Here we are using as a protoplast cells. We are embedded with the agarose. So agarose embedded with our protoplast are poured into the liquid medium. So these cells are able to grow, but it's having importance that this technique is mostly used for extraction of secondary metabolites. As we know, the primary metabolites which are formed in the cell are utilized by plant for its growth and development, but the secondary metabolites which are formed by the plants are excreted out. So in case of agar beads or agar droplet beads, when these cells are growing, so they are secreting these secondary metabolites into the medium. So this is the importance of using protoplast in a bead form. The another technique which we are using for protoplast regeneration is the microisolation. When we are having very sensitive protoplasts which are very friable in nature, that time we take only single protoplast, fused protoplast, and place it on a small volume of medium. We take single protoplast along with the small volume of medium in a petri plate. Then we seal this petri plate with the paraffin. So when this protoplast will be able to grow and divide and form the callus, then from this isolation we can take this cells into the liquid medium where we will be able to form maximum mass of callus. Then this callus is again subcultured. Then from a single cell we will be able to form mass of callus or n number of plants. So last technique we are using for regeneration method is co-culturing. What we do in case of co-culturing, we culture two different protoplasts together in a liquid media. But why we do so? First, we are taking cells which are actively growing, or hybrid protoplast, which are actively growing to form its cell wall, and these are always healthy and viable protoplasts. Along with this vigor hybrid protoplast, we take another protoplast which are from those plant sources which don't have high growth rate and their genotype varies. These low survival hybrid protoplasts are grown with the high survival protoplast in a same culture. It has been observed that culturing of high growth with the low growth protoplast in a same medium help to grow these low growth protoplast culture in a liquid medium. In a simple language we can say, हम लोगों ने we are taking in co-culture, we are taking two protoplasts, protoplast A and protoplast B. In case of protoplast A, what we are doing, we have taken those protoplasts, hybrid protoplasts, which are actively growing vigorously, and they are all viable. They are able to form cell wall within in the 24 hours. Then they will form the callus. So this is the specification of the species A. Now the species B, which have protoplasts, are taken from those plant source which are not dividing fast, which are not having good genotype, but due to having some importance in their genes, we were able to grow this protoplast, or we fuse this protoplast with the another species of the हम लोग स्पीशीज ए लिया, बी में क्या लेते हैं कि हम लोग हाइब्रिड प्रोटोप्लास्ट उनसे लेते हैं जहाँ पे जो एक्टिवली ग्रोइंग सेल्स नहीं होती है. What they have observed by culturing highly vigorously growing protoplast with the less vigorously growing protoplast, so they have observed that जो A का इफेक्ट है वो B पे भी पड़ता है. Then these species B also are able to grow under in vitro conditions. So means ये एक दूसरे को क्या करते हैं, हेल्प करते हैं. They help each other in their viability in the formation of cell wall as well as the callus formation. As we know, when the techniques are done, now the first step in the plant regeneration is the formation of cell wall. So we place it our hybrid protoplast in any way. Now the first step is the formation of cell wall. So what we do, as we know, the plants are these hybrid protoplasts are able to generate cell wall within in the 24 hours. So the division, cell wall division or the callus formation depends on from which variety of plant species we have taken our protoplast. First, second, genotype of that protoplast. Third, protoplast source, from which part of the plant we have isolated protoplast. Then, which procedure which we have used for isolation of protoplast. Then the last, composition of the medium. All these parameters work together for the cell wall division and callus formation. It has been observed that the plant material which has been taken from embryonic calli, if we talk about the embryonic calli, it means the callus formed by the embryonic cells. It means we have taken this plant material in taken from in vitro condition, or we can use cells from cell suspension culture. We can use embryonic calli or we can use suspension culture cells to isolate the protoplast. These both embryonic calli as well as cell suspension culture are having ability to generate or to divide fast. If we will compare these two X plants with the mesophyll cells or the plants taken from the greenhouse, they are having less efficiency to develop cell wall under in vitro conditions. Now the critical factor in this cell wall formation, we have to maintain the plating density, means how many protoplasts should be present in a liquid medium. So many scientists have worked together. They have observed that 5.0 into 10 raised power 4, 5.0 into 10 raised power 4 up to 1.0 into 10 raised power 5 protoplast per ml can be maintained. Again, I will repeat. To maintain the plating density of the protoplast, we can maintain density from 5.0 into 10 raised power 4 up to the 1.0 into 10 raised power 5 protoplast per ml. So this plating density has to be maintained. If we will keep our plating density less than this value, then we won't be able to get viable protoplast. Second, the protoplast will not be that vigorously growing and their viability will get decreased. Or if we will increase our density than this above said value, then there will be overcrowding of the protoplast. So the hybrid protoplast will not be able to grow properly. So again they will decrease the viability of the protoplast cells. Now the another factor which play important role in the cell wall formation is culture medium. As we know till now, B5 and MS media play important role in the development of our hybrid protoplast. It has been observed that there should, if there is ammonium ion present in the medium, that hampers the development of protoplasts. So there are some protoplasts, hybrid protoplasts, which are having sensitivity to the ammonium ions. So while choosing our protoplast from different species, we have to be sure the protoplast which we have taken is ammonium sensitive or not. So another factor is their osmoticum. As we are having many osmoticums there, sucrose, mannitol, sorbitol or glucose. As I have told you that mannitol or sorbitol are seen best as a osmoticum, but it's not applicable to all the plant species. Like for potato, cassava, sweet potato, we use sucrose as a osmoticum. We use sucrose as a osmoticum. As the protoplast of potato, cassava, or sweet potato develops, these sucrose get utilized by the protoplast cells, and the osmotic pressure of the medium get reduced by its consumption. So as cell grows, the osmotic pressure will get automatically decreased because these osmoticums are metabolically active. So we have to maintain these protoplasts under the dark conditions, as there are many protoplasts, or it has been seen that most of the protoplasts are sensitive to light. So what we do, we will keep our hybrid protoplast under, or incubate our hybrid protoplast under dark condition. Till that time, they will be able to form cell wall. At what temperature we have to maintain our hybrid protoplast? Mostly in the range of 25 to 30 degrees centigrade. So when the protoplast has developed its cell wall, now after cell wall they will start division. The first report of plant regeneration was observed in Nicotiana tabacum. As we know, the Nicotiana tabacum, most of the research has been carried out on the Nicotiana tabacum, and this regeneration power of Nicotiana tabacum was reported by Takabe et al. In 1971. Since then, from 1971, it has been observed that these protoplasts are having regeneration power. So they have using many species, many genera, and many plants families have been used to get the hybrid protoplast. Again, if we will talk about the plant regeneration capacity, how we will decide the plant regeneration capacity, again we can say the plant regeneration capacity depends on the medium which we are using for our hybrid protoplast, genotype of the species which we have selected for protoplast isolation, and the next we can say that the growth regulators which play important role in the development of protoplasts. So